Yes, Mr. Hill.
We are going to continue today, sir, with the presentation on the Highland and Travenol products. When I finished on Friday, I was just about to move to Haemophil M, and it's perhaps worth reminding everybody what the product was. It was described by Dr. Kingdon in his draft witness statement as being a product produced as a result of a solvent detergent method, which had been licensed from the New York Blood Center by Highland Travenol. He described it as a freeze-dried, high-purity concentrate in which lipid-enveloped viruses were dissolved using the NYBC process prior to fractionation. The process was adopted in order to inactivate non-A, non-B hepatitis in the plasma pools. That is the reason why that method was used. If we could have on screen, please, Shumik, SHPL 40409_072. Let me try again, SHPL 40409_072.
409, did you say?
Yes, sir, SHPL 40409.
Yes, I think the O in 409 got missing.
So it appears that there seems to be a technical glitch and we can't bring the actual document up, but I think I can describe it to you without presenting it on the screen. It is a memorandum, an internal...
Well, just pause for a moment. Is it a technical glitch which affects only this document, or is it a more general one?
We checked the documents this morning and they all seemed to be present.
It's there, but I think the document is corrupted.
I think we think this document is corrupted. So
it's there in the system.
Right.
Can I just try the next document that we're going to go to and see whether or not this is a more widespread problem? Let's try SHPL 40293_141.
Right, we'll just take a break.
Thank you, sir.
Right, let's start again.
Can we have on screen, please, Shumik, SHPL 40409_072, please. This is an internal Travenol Laboratories document dated the 28th of August 1987, and it concerns the crossover between Haemophil T and Haemophil M. It says the following with reference to a previous memo. Highland believed that FDA license could be issued by the end of November 1987. We understand that to be a reference to an FDA license for Haemophil M. Hopefully, UK customers will commence usage on a named patient basis.
And I'm missing something, am I? Just where does it say?
So this is there's a problem with the referencing again. Shunik, can we have SHPL 40409_072? Yes, sorry, I brought up the wrong document there. So 20th of October 1987, an internal Travenol document, Travenol Laboratories Limited, the UK company, with reference to a memorandum of the 12th of October. Highland believed that FDA license could be issued by the end of November 1987. Hopefully, UK customers will commence usage on a named patient basis once M has FDA approval. Premium price makes this very limited. Haemophil T, heat-treated, that is the product that we were looking at on Friday, not accepted by marketplace, poor solubility and record on viral safety. Armour, major competitor as monoclonal productions, do not have FDA license either, but the marginalia shows that that license was granted after the memo had been typed. In view of the above, we must submit our license application without delay once data collated. We can see there's, again, there is going to be reliance placed on the named patient basis, at least until a license is obtained. On the 7th of December 1987, thank you, Shumik, that can be taken from the screen. On the 7th of December 1987...
Can you just help? Do we know any more about the record on viral safety that's referred to there, poor record on viral safety?
I understand that to be a reference to the transmission of non-A, non-B hepatitis, which by 1987 it was recognised that haemophil T was still transmitting non-A, non-B hepatitis.
But being a poor record which was not accepting, the marketplace not accepting it, would mean that other products by rival competitors were better, presumably. Presumably, yes. The marketplace seems to have reacted not just to the lack of the good record on viral safety, but also the solubility of the product. Yes.
But certainly by 1987, there was an acceptance that 8Y, the NHS product, for example, was...
Well, I follow that that's so in the UK. Is this memo just talking about the UK? It talks about FDA approval.
It does, but it is an internal Travenol Laboratories Limited, so a UK company, memorandum. The FDA, the reference to the FDA, I understand to, in this context, to be useful to them in order to market the product in the UK by pointing to the fact that the FDA have licensed it.
Yes.
On the 7th of December 1987, Highland and Travenol Limited applied for a clinical trial exemption for haemophil M in conjunction with Dr. Savage at St Thomas's Hospital. That was granted on the 20th of January 1988 for a period of 3 years. That's SHPL 40496_191. That, of course, just a few months after that memorandum that we've just been looking at. The application for the full product license was made on the 12th of June 1989, so some 18 months after that memorandum. The reference is SHPL 40496_149. The application was initially refused on regulatory grounds as it was considered to be a high technology product under European law and consequently subject to a separate licensing regime, which the product then went through. The license was eventually awarded in 1994. I'm not going to go through all of the detail in respect of that. I will give the references to the original application, which is SHPL 40496_149, then SHPL 40293_182, and SHPL 40468_064. Those are the main documents which show what happened to that application. Of relevance to the inquiry is the fact that the product was still available in this period between 1989 and 1994 when the licensing process was going through its motions. It was still available on a named patient basis. A DHSS note dated the 14th of November 1989 said that Baxter were supplying approximately 2 million international units of their unlicensed product to the UK each year. We understand that to be a reference to haemophil M. The reference for that is DHSC 302412_077. Shumik, could we have on screen, please, SHPL 40293_141. Page 2 of that document, please. This is a document from the 21st of May 1990 from Patrick Rafferty, who was medical director with Baxter to Ron Feeks at Thetford. So it's an internal memorandum. By this stage, the company's name has changed from Travenol Limited to Baxter Limited. Baxter Healthcare Limited, sorry. What Dr. Rafferty says in the memorandum is this. In my capacity as medical director, in order to understand more fully the clinical management of haemophilia patients and the potential role for highly purified products in the centre of their hemostasis, I have met with most of the haemophilia centre directors in the past few months. Following these meetings, I have received a number of inquiries relating to the effects of monoclonally purified factor 8C on the levels of hepatitis C antibodies in patients. It would appear that there is a relationship between the progression of hepatitis cirrhosis and the levels of hepatitis C antibodies in some patients. If this relationship exists, and if monoclonally purified factor 8C is confirmed as being effective in significantly reducing or eliminating the levels of hepatitis C antibody in these patients, then as medical director, I have a moral obligation to bring this to the attention of the clinical experts who are responsible for the management of haemophilia patients. My understanding is that the Baxter monoclonally purified factor 8C, Haemophil M, is currently not yet licensed in the UK. Perhaps you could advise me on the most ethical approach for me to take in this regard, whilst meeting appropriate regulatory requirements. We have a response to that memorandum.
Just one question on this. He doesn't say what the relationship between the level of hepatitis C antibodies and the progression of cirrhosis is, whether it means that the more antibodies, the less cirrhosis, or the less antibodies, the less cirrhosis.
No, he doesn't. What I take from this memo is the view of Dr. Rafferty that Haemophil M is an effective product to use to limit the risk of hepatitis cirrhosis. And the way that Dr. Rafferty expresses himself is that he feels then that he has a moral obligation to share that information with the haemophilia clinicians.
And why is he then raising that? Plainly, as a commercial sales point, it would seem sensible to highlight it, but the constraint might it, that would then fall foul of the advertising prohibition on a named patient basis.
Exactly so, sir. And that is what is said in the reply to
I see.
to Dr. Rafferty's letter.
So, where does that leave his moral obligation? To break the law, or what?
So that is a question for Dr. Rafferty.
Yes.
If we could have on screen, if we look at the reply, we will see the position that was taken by Mr. Feeks in response. It's SHPL 40293_142. The date is 1st of June 1990, and it's a response to the memorandum we've just looked at of the 21st of May 1990. What Mr. Feeks says is this: the area of personal representations and unlicensed product is one where great care must be taken in order to avoid breaking both the acceptable code of conduct, e.g. that of the ABPI, and the law itself. The Department of Health has, in the past, prosecuted companies for advertising unlicensed products. Part 6 of the 1968 Medicines Act on the promotion of sales of medicinal products clearly indicates that only licensed products can be promoted, and that the spoken word by company representatives constitutes a form of promotion. Moreover, the legislation under which we import Haemophil M, SI 1984-673, states that the importer, quotes, will not at any time issue or cause another person to issue any advertisement or make any representation in respect of that medicinal product, and that he will only sell or supply that medicinal product only in response to a bona fide unsolicited order. Thus, your actions must be very carefully calculated, such that you cannot be accused of promoting Haemophil M. This is doubly important, as Baxter received a formal complaint from the DOH, the Department of Health, in early 1988 concerning our stand at the ISBT meeting held at Wembley late 1987, brackets promotion of GammaGuard. Further transgressions could be very serious indeed. To give you a framework in which to operate, you must abide by the following rules. 1. The provision of factual scientific information in response to a request from a physician does not in itself constitute promotion. Therefore, you should not offer such information until requested. 2. You should not approach individual physicians with claims of safety for an unlicensed product. Such information must be made available through the scientific press by publication of data and substantiation by peer review. 3. You should not set out to derogate other licensed products. 4. Discussing the therapeutic value of haemophil M with a physician is only acceptable if it is the physician and not you who initiates the conversation. Although this does limit you quite substantially, you should abide by these guidelines, otherwise we could, at the very least, antagonise the Department of Health. This might in turn be detrimental to our product licence application. Going on to the availability of haemophil M, when asked, it is quite acceptable for you to inform the physician: 1, we hold small stocks in the UK. 2, the product is available on a named patient prescription basis. 3, the cost of the product. 4, contact names in marketing for further information, and regulatory for prescription release details. That is Mr. Feek's response to the conundrum that has been presented.
Can we just go back to the first page of this letter? Just looking at the legislation, will not at any time issue or cause another person to issue any advertisement or make any representation. So what this might mean is it that he couldn't tell the DHSS, for instance, of the claims he was making for the suitability of haemophil M, with the view to influence them letting the profession know, because that would be effectively causing another person to make a representation.
That would certainly be an interpretation that would be open to a court.
Well, it would be straight within the language
Yes.
that's quoted there, at any rate.
But there is what Mr. Feeks says as one of the solutions to this is to publish your information
In a scientific journal.
in a scientific journal. Sharing pre-publication information with the DHSS, I don't think would fall foul of this, unless there was an intention that the DHSS then go on to inform and make an advertisement or a representation to various clinicians.
Well, it's difficult to think of what other interpretation, what other reason there would be.
There may be a reason to begin the discussions which lead to a product license. For example, discussing how the license is best approached, what form of licensing should, what format the application should take, whether it should be a rich license and so forth.
I see that might be a possibility.
But certainly it wouldn't be open to the company to try to work their way around the prohibition by using official channels.
Thank you.
I don't intend to say anything further on haemophil M. Turning to Proplex, this was the freeze-dried concentrate that contained factors 2, 8 and 10, as well as factor 9. I don't intend to go through the licensing story for this product. I note only the following, that interest in the product in the UK can be traced back to at least August 1974. That's OXUH 40630. Dr. Biggs there appears to have obtained some for emergency use on a named patient basis. In March 1975...
Do you have that reference again?
OXUH 40630.
630, thank you.
In March 1975, a temporary license was granted to import the product, that is MHRA 0033317_093. The application for a product license was made on the 21st of May 1975, MHRA 0033317_095. The license application was considered by the CSM Subcommittee on Biologicals in November 1975. So this is a month before the World in Action film. The medical assessor at that time commented that Proplex would appear to have been adequately manufactured, but is subject to possible contamination with hepatitis virus. But the medical assessor recommended that, subject to approval of quality control, a license be granted. The subcommittee concluded that a product license be granted subject to several conditions. And it's perhaps worth looking at that document. MHRA 003317_079. MHRA 003317_079. You can see November 1975, Subcommittee on Biologicals, a month before the World in Action film, or, well, the previous month, we don't know how many weeks exactly. On the evidence before them, the Subcommittee on Biologicals recommend the grant of a product license for this preparation, provided that information is provided on the number of donations from which the plasma is pooled for the manufacture of prothrombin complex, and 2, the reasons for and rate of rejections of donors and donations centre by centre. There are then some further provisos that I won't take you through. If we could have on screen now, please, Shumik, MHRA 003317_077. This is a response from Travenol Limited to a letter that has been sent by the licensing authority on the 29th of January 1976. We don't have that letter. This is the response to it, addressed to Dr. Andrews, the Senior Medical Officer at the Department of Health and Social Security. It provides further information that has been requested as part of the licensing process. It says 1A1, the size of plasma pools used for the manufacture of each lot of factor IX complex can vary up to approximately 6,000 litres. 1A2, the reasons for rejection of a donor or a donation are outlined in the product license application, Part 3, Section A. Donors are most commonly rejected due to serum protein electrophoresis unacceptability, or for being positive for hepatitis B surface antigen, or on the RPR test. Rejection for either the first or last reasons given above would normally only be for a limited period of time. We can see in the marginalia on the right-hand side is written no rate of rejections. That information was requested by the DHSS but hasn't been provided in this letter. We don't know, sir, whether that was provided subsequently or not. We haven't seen any information, but it was. That does not mean that it was not. Proplex was granted a license on the 15th of October 1976, SHPL 00000232_001. The timing is interesting, sir, in that the original consideration by the CSMB fell before the World in Action documentaries were broadcast. The letter that was sent in January was sent afterwards.
What was the information about that was requested ever actually provided, so far as we know?
We haven't seen any information that it was, but we have rather incomplete documentation from that period, so we can't say that there wasn't another letter that was followed up. Certainly, somebody seems to have spotted that no information has been provided on the rate of rejections, but we don't know whose writing that is.
Yes.
The 1978 Haemophilia Centre annual returns show that the compiled information from 103 centres. Of the 2.2 million units used by haemophilia centres to treat haemophilia A patients with antibodies during 1978, only 149,000 units were Proplex, and that compares with 1.9 million units of FEBA. So we can see that although Proplex is used, it is far less popular than FEBA for inhibitor patients. It's not clear from the information in the returns how much was used for factor IX patients. I won't go through the subsequent history of the renewal applications and considerations. We can return to that if necessary. The picture is not always clear as to when or indeed whether renewal applications were considered, but nor is it clear the extent to which Proplex remained on the UK market during the 1980s. And that completes what I have to say about the licensing of the Travenol products. I'm going to turn now to the communication of risk associated with those products. We have seen some indication of this in the documents that we've looked at, but this is to try to fill out the narrative a little. If we go back to Haemophil and the original version of Haemophil, which was the non-heat treat Haemophil, as we have heard, the product license application was made in November 1972. A label was sent with that application, which contained the words, "Warning, the risk of transmitting hepatitis is present." The package leaflet was also contained in the application. And could we have this on the screen, please, Shumik? It's SHPL 40275_013. We can see this is the part of the application, and it's the draft package leafleting. If we go over to the next page, please, Shumik. Actually, 2 pages on to the 5th page of the document overall. Under the heading Cautions, it says, this concentrate, this is the second paragraph down, this concentrate is prepared from large pools of fresh human plasma. Such plasma may contain the causative agents of viral hepatitis. There is no known laboratory test to demonstrate either the presence or the absence of such agents, and the concentrate has not been subjected to any treatment known to diminish the risk of transmission of hepatitis, since such treatments greatly increase the loss of AHF activity during preparation. The concentrate should, therefore, be used when its expected effect is needed in spite of the unknown hepatitis risk associated with its use. Special consideration should be given to the use of the concentrate in newborns and infants, where a higher morbidity and mortality may be associated with hepatitis. You may recall, sir, from Friday, the comment made by Dr. Thomas, the medical assessor at the time, that in the product application, there was no attempt made to disguise the risk of hepatitis associated with the product.
Just looking at that for a moment, the 5th line down, starting the end of the 4th line down in the 2nd paragraph, and the concentrate has not been subjected to any treatment known to diminish the risk of transmission of hepatitis, since such treatments greatly increase the loss of AHF activity during preparation, which is interesting. phrase because it suggests, A, that the company has considered treating the product, B, that it is not saying it can't be done. It is saying it's uneconomic or it lessens the amount of factor VIII you get out of the product, not that you get none.
Yes, sir.
So it could, on that interpretation, on the words themselves, mean that they could produce a safer product, does it? Only it's just not worth the candle.
I'm reluctant to try to interpret what was meant from the warning label without
The reason I raise this is whether there's any other document that you know of that sheds any light on that.
Not that I'm aware of, but it is something that we can continue to look into. I'm not sure what methods of viral inactivation were being used or being thought about by the company as of 1972.
Well, whatever they were, that sentence is at least open to that interpretation. Is it better open to another one or not?
It is open to that interpretation, yes.
Yes. Thank you.
The license was granted in February 1973, as we heard on Friday. A package leaflet dated from 1979 contains a broadly similar warning, but with some further information about screening tests. It may help to have a quick look at that. SHPL 301055_023, please, Shumik. This is, as I say, dating from 1979, and we can see in the warnings section, this concentrate is prepared from large pools of fresh human plasma. Such plasma may contain the causative agent of viral hepatitis. However, each unit of the plasma used in manufacture of this product has been found to be non-reactive for hepatitis B surface antigen by radioimmunoassay. The concentrate has not been subjected to any treatment known to diminish the risk of hepatitis transmission, since such treatments greatly increase the loss of AHF activity during preparation. The concentrate should, therefore, be used when the need for its expected effect outweighs the hepatitis risk associated with its use. This lot, after reconstitution for use, has been found non-reactive for hepatitis B surface antigen using a solid-phase radioimmunoassay technique licensed by the US Bureau of Biologics. The significance of a non-reactive test result with concentrate antihemophilic factor has not been established. Therefore, the product should continue to be considered to carry a risk with respect to hepatitis. Turning to haemophil, I should add so that we have less information about the data sheets for haemophil than we do for the immunoproducts that we looked at last week. That's just the nature of the type of sources that we have to work with. Turning to heat-treated haemophil, we heard on Friday that at the time that the original application for heat-treated haemophil was considered by the Committee on Safety of Medicines, Subcommittee on Biologics, it was, quote, strongly deprecated that leaflets had been sent to doctors, which the committee considered to be promotional leaflets making unjustified claims. I won't go back to those. A further application was made on the 30th of November 1984. The proposed bottle label contained the words, quote, warning, the risk of transmitting hepatitis is present. The carton label contained the following is SHPL 40283_005. Please, Shumik, it's page 24 of that document. in the section headed Warning, a little further down the page, plasma from which this product was derived was found to be non-reactive for hepatitis B surface antigen, HBsAg, when tested with licensed third-generation reagents. In addition, the process used in the manufacture of this product includes a heating step designed to minimize the risk of transmission of hepatitis. However, no procedure has been shown to be totally effective in removing hepatitis infectivity from antihemophilic factor human. The proposed direction sheet contained a similarly phrased warning. It's at page 30 of the same document. I won't go to it, but it also included the comment, quotes, although these testing and heating steps reduce the risk of hepatitis transmission, the possibility of such transmission should be considered in the use of the product. The data sheet had a similar warning. That's at page 40 of the same document. And the direction sheet contained a summary of the understanding at that time of the chimpanzee trials, to which we referred on Friday. That is at page 29. The amendment to the licensing haemophil T was granted on the 27th of February 1985. The particulars of the license included a warning in the same terms as those contained in the proposed direction sheet and data sheet. That is MHRA 5087. It's page 14 of that document. We also have a carton with the expiry date June 1988, which shows the same wording as the sample carton that was included in the application. That's SHPL 0001013_004. You'll note, sir, that in those warnings there is no reference to HIV, LAV, HDLV3, although, as we heard, Dr. Kingdon's view was that haemophil T did inactivate HIV. Haemophil M, as we've just heard, not licensed until 1994, but available on a named patient basis seemingly from 1987. I'm not going to take you through the labels that it contained, but we can, for the transcript, the warnings contained in the original application are at SHPL 40375_082, and in the subsequent revised application SHPL 40396_003. Proplex, the application for the product license was in May 1975, as we've heard. I won't take you to the documents, but the proposed direction sheet and data sheet contain a similar warning to the contemporary warnings for haemophil at that time. The reference is MHRA 0033317_098. I will, if I may, sir, take you to a data sheet from February 1983 from Proplex, SHPL 40963_002. If we look in the central column at the bottom, we can see it's revised in February 1983. And this has been printed by the US company, Travenol Laboratories, Inc., and printed in the US, as it states there. If we could go to the next page of the document, please, Shumik. Under the heading Warnings, the second paragraph again. This product is prepared from large pools of human plasma. Such plasma may contain the causative agent of viral hepatitis. Although each unit of source plasma used in the preparation of this product
has been found to be non-reactive for hepatitis B surface antigen by licensed third-generation reagents. The concentrate has not been subjected to any treatment known to diminish the risk of transmission of hepatitis. The product should therefore be administered only when its expected effect outweighs the hepatitis risk associated with its use. If we go down a couple of paragraphs lower, under precautions, just above the subheading laboratory tests, you see a further warning, which is, and it's the last paragraph there, special caution should be taken of the use of this concentrate in newborns, where a higher morbidity and mortality may be associated with hepatitis, and in individuals with pre-existing liver disease. Again, terms similar to those that we have looked at from the 1970s for haemophil in a document from February 1983. It's not clear on the face of this document whether or not it was a document that was used with UK product, but that is what the American company was printing at that time. A version of the leaflet was revised in October 1984. The reference is SHPL 0001049_034. The same warning was issued, and again, there is no reference to AIDS, HDLV3, LAV, HIV. That is from October 1984. Once more, it's not clear from the face of that document whether or not it was used in the United Kingdom. That is all I intend to say on the communication of risk in respect of the Travenol products that we have been looking at. And I will turn now to the question of donors. In terms of donor pool sizes, we've heard evidence already on this, and I won't go back to it, but I will just remind you, sir, that Dr. Thomas's summary of the report produced following the inspection of the Highland blood banks, which we looked at on Friday, said that donor pools contained plasma from as many as 6,000 donors. That figure was confirmed in a letter from Travenol Limited, dated the 22nd of June 1976, about Proplex factor 9, which we looked at earlier today in the licensing section. The reference is MHRA 003317_077. The reference for Dr. Thomas is DHSC 0105593 _006.
Thomas was talking about 6,000 donors, did you say? What you showed me earlier talked about donations.
Yes. There is that distinction between and donations. Whether or not it was an intended
in fact, litres, I think it was, wasn't it? 6,000 litres is what you mentioned.
Yes.
And that's what MRHA 232001, I think, or SPHL 232001. If it's 6,000 litres, that's about 9,000, 10,000 donations, isn't it?
Depending on the level.
Assuming that a donation of plasma is about 600 and odd millilitres.
Yes. It's not always clear in the documents whether references to donors are intended to specifically mean donors, or whether it is intended to mean litres of plasma or donations. But there's a lack of precision sometimes.
Well, it does matter.
It does.
Because if you take, on one view, any risk per donation is 1.5 times as much if you use different measure.
Yes. What we know from Dr. Kingdon and his witness statement that we looked at on Friday is that he referred to plasma pools of up to 15,000 donations.
Yes.
He was very specifically talking about donations there. So whether or not 6,000 donors or 6,000 donations was meant in circa 1975, by the time of Dr. Kingdon's statement in 1990, there appears to have been a rise in the start pool size up to 15,000 donations.
Unless there's other evidence to help interpret, one just has to take the words as they come and interpret them on that basis.
Yes, sir.
I think. Has more?
Yes. Yeah. But I think that the evidence does show that there was a growth in the size of the donor pools used
Yes.
by Travenol. Turning to the question
So
of...
the growth that you would identify would be, what, 6,000 up to?
yes.
6,000 litres or donors, depending on which,
Yes.
up to 15,000 donations.
Yes. We don't have any further detail as to when, why, and with what justification.
Or whether by steps, etc.
Exactly. We can seek to try to find out more, but I'm not sure how much we will be able to find out on that. Turning to the procedures that we used for the donors, we heard from Dr. Kingdon's statement his evidence about how things were done. The statement was dated 1990, and it was sometimes unclear which period he was talking about in that statement. And out of fairness to Dr. Kingdon, it's important to note that he joined the company in 1981. So some of what we're going to look at now took place before his time, and he wouldn't necessarily have known about it. But if we could have on screen, please, Shumik, SHPL 40279_012. This is a document which is part of what is called the Donor Centre Technical Guide. It's published by Highland Division of Travenol, Inc., so the American company. We can see that the manual is dated in the top left-hand corner box, March 25th, 1975. I note that is before World in Action documentaries were broadcast, but that doesn't tell us when the documentaries themselves were actually filmed. We don't know which part of 1975 they were.
A revision issued on the 30th of April 76.
This is, the original manual was dated March 25th, 1975. This is the second revision to it. And it's April the 30th, 1976, which is after the World in Action documentary. And it says, donor requirements checklist. Purpose, to establish the instruction regarding a guideline checklist of requirements to be followed in determining suitability of a donor to qualify for whole blood or plasmapheresis donations. Scope, provision of this instruction applied to all Highland plasma centres. We can see then a list of instructions. We don't have all of these documents, but you can see the types of area that the different instructions cover. The fingernail dye examination and fixation, which I understand to be a method to try to prevent people donating too frequently. The weight, temperature, blood pressure, hepatitis B surface antigen tests, serological tests, and physical examination. As I say, we don't have all of the documents that were part of that guide at that time, but we do have some documents which formed part of the guide over the succeeding years. If we could have SHPL 40279_031, please, Shumik.
The original variant of this instruction was March the 30th, 1976. This is the 5th revision, dated May the 11th, 1976. And again, we can see it's part of a donor technical guide. Purpose: to establish the instruction for maintaining adequate permanent donor records. We can see the types of information that are required in that record, including the donor's name, permanent donor number, date of donation, bleeding number, vital signs, and then the result of the syphilis test is to be included. The donor record form is to be reviewed by the attending physician at least once every 4 months to determine continuing suitability of the donor. I won't go through the rest of the document, but we can see what the instruction says there. If we could go, please, Shumik, to SHPL 00000279_029. This is a information sheet about establishing the instructions and the use and proper documentation of source plasma (human) labeling. The original document from 15th of December 1975. This is the 3rd revision, dated 29th of October 1976, and it sets out what should be contained on the label of the plasma, which should be affixed to every plasma container. That includes the donor name, the donor number, the bleeding date, and so forth. Then on the next page, we can see that procedures are put in place for each label to be accompanied by 16 smaller labels that can be used for the identification of plasma segments for testing, donors' bleeding bags, documentation on donor records, summation reports, Vacutane tubes used for special programs, samples for laboratory testing, and donor center logs. Shumik, could we please have SHPL 40279_018? This instruction is about establishing the proper documentation of the whole blood and plasma processing log. The issue date is the 18th of January 1977. It is the 2nd revision of a document that dates from the 6th of October 1976. We can see the instructions given about the information that should be recorded, again, including the bleeding number of the patient. I won't go through the large number of other similar documents, but we can see from them that there was a technical guide which contained such instructions. Obviously, what the documents don't tell us is how those instructions were then implemented on the ground. Jumping forward to October 1983, if we could have on screen, please, BAXT 5011_002. This is a document which is entitled Highland Division of Travenol Laboratories Limited, file RA1002, October 1983, source plasma, human, and it is produced by Travenol Laboratories Limited, so the UK company rather than the US company. If we could turn over to the next page, please, Shumik. The contents are set out there. The 4 sections to it. The first section is about product description, donor definition, and facility requirements. The second section is the donor center technical guide, table of contents, and that is a reference, as I understand it, to the same guide that we have just been looking at, but this is the version which is now in place in 1983. Part 3 is the establishment licenses and lists of approved plasma collection centres, and Part 4 is some of the
regulations from the United States. So it appears to be a document that the UK company has produced to inform itself or others of the way in which the American company goes about collecting its blood, plasma, and whole blood. If we could turn to page 5 of that document, please, Shumik. I'm not going to go through it all. It is quite lengthy, but just certain features that can be picked out from it. 2C, each unit of source plasma, human, shall be non-reactive for hepatitis B surface antigen when tested by a suitable test system of third-generation sensitivity. 2F, a label shall be affixed to each unit of source plasma, delineating the following information. It gives the information that should be on the label, including the donor number. If we go over to the next page, please, Shumik. This is the section about the suitability and safety of donors. And it says, the suitability of a donor shall be determined by a qualified licensed physician or by persons under his supervision and trained in determining donor suitability. We can see, then it goes on to describe the method of selecting the donor. Informed consent, then the medical examination, which says this. Each donor shall be examined by a qualified licensed physician on the day of the first donation and not more than 1 week prior to the first donation, and shall be certified to be in good health by the examining physician. Medical examinations shall be performed at subsequent intervals of no longer than 1 year. It then goes on to list the qualifications of the donor. There is an age qualification, a weight qualification, a qualification for blood hemoglobulin, a requirement, if we go on to the next page, please, Shumik, of hepatitis B surface antigen freedom. If we go down to N, freedom from any disease other than malaria transmissible by blood transfusion insofar as can be determined by history and examination indicated in this section. O, freedom of the arms and forearms from skin punctures or scars indicative of addiction to self-injected narcotics. And P, freedom from a history of viral hepatitis. The general section states, any donor who, in the opinion of the interviewer, appears to be under the influence of any drug, alcohol, or for any reason does not appear to be providing reliable answers to medical history questions, shall not be considered a suitable donor. Then continued donor suitability. The accumulated laboratory data and collection records of the donor shall be reviewed by a licensed qualified physician every 4 months to determine and carefully certify whether or not the donor may continue on the plasmapheresis programs. So that is the section about the suitability and safety of donors. And you may find interesting, sir, the distinction made between when a licensed physician has to be involved and when somebody operating under the supervision of that licensed physician is involved. Supervision is not, so far as I'm aware, defined in this document. If we could go, please, to page 10 of the document. Donor identification system. It says a donor identification numbering system should be established that positively identifies each donor and relates such donor directly to his blood and his plasma, as well as to his accumulated records and laboratory data. Such systems shall include either a photograph of each donor, which should be used on each visit to confirm the donor's identity, or some other method that provides equal or greater assurance of positively identifying the donor. I won't go to it, but at page 15, there is a further detail on the donor records that are required. Page 18, please, Shumik. I'm not going to go through this, but we can see there that it is a summary of the FDA licensing procedures that are in place as of 1983. We then see on page 19, please, Schunig, the table of contents from the Donor Centre Technical Guide, so the iteration of the document that we've been looking at previously. And if we could go on to page 20, please, Schunig. I won't go through all of these, but we can see a large number of instruction sheets which form the guide. It goes on for some 6 pages. And then, please, Schunig, if we could go to page 29. This is the list of centres that are used by Highland. And it says that plasma used to manufacture Highland blood products is collected only at plasma collection centres licensed by the United States Food and Drug Agency. The centres and their locations are as follows. Refers to the Highland Therapeutics Division in Glendale, California, and Travenol Laboratories, Inc. in Deerfield, Illinois, and then a list of locations, all of which appear to be in the United States. If we go over to the next page, please, Shermick. As I say, all of these appear to be in the United States. There is one reference about a third of the way down to Cherry Street Plasma Center, Inc. There is no additional information provided as to which city that is in or which state that is in, but all of the others on the list are in American cities, in American states. I don't know if there is any significance to the lack of information about the Cherry Street Plasma Center. As we saw from the contents page, the document also contains extracts from the US Code of Federal Regulations concerning food and drugs, but I'm not going to go to those. We can take that off the screen. Thank you, Shermick. So that, sir, is the documentation that I wish to show you about the internal processes, at least as written, by Highland and Travenol at the time, 1970s and 1980s. I would like to turn now to the issue of the use of plasma from prisons. Could we have on screen, please, Schumick, CGRA 40291. Some of this material, sir, is are documents that we will come back to in November when we're dealing with the way in which the pharmaceutical companies responded to the risk of AIDS. My focus today is on how plasma was being collected before circa 1983, when the risk of AIDS becomes increasingly apparent. So we will turn back to this material at a later stage for a different purpose. This document is dated the 20th of October 1983, and it is an internal memorandum within Travenol, and it is sent from W.R. Srigley to D. Castaldi about a telecon with Dr. Donahue. Now, Dr. Donahue is Dr. Dennis Donahue of the FDA's Division of Blood and Blood Products. And it is around this time that he is speaking to pharmaceutical companies about the risk of AIDS and what they are doing about it. And what is attached is stated to be the text which I used to define for Dr. Donahue's Travenol's policy with regard to the use of plasma from prison centres. He expressed his satisfaction with the policy and appreciation that we responded promptly to him. He asked that we not send it to him in writing at this time.
If you have any additional questions regarding the conversation, I would be happy to discuss it in detail with you. So it appears to be the text of a policy that was explained orally to Dr. Donoghue about what Travenol were doing in respect of plasma from prisons. If we go to page 2, you see the document is headed Proposed Text for Conversation with Donoghue, but we can see from the memorandum that this is the text that appears to actually have been used in the conversation. What it says is this: we had previously made the decision to discontinue the purchase of plasma from licensed centres in prisons. To that end, we have chosen not to renew any pre-existing contracts with such centres after this year. Following my conversation with you yesterday, we have decided that we will promptly discontinue the use of such plasma for the manufacture of coagulation factors. We have pools in process this week which contain plasma drawn in prison centres. As of the end of this week, we will not make any coagulation factors from any of the prison plasma which remains in our possession or which we are contractually bound to purchase through the remainder of the year. No further plasma pools used for coagulation factors will contain prison plasma. Any intermediate fractions manufactured from plasma pooled this week or in previous weeks would be processed to final product. That is what the company, and I remind you, sir, it is the American company, is telling the FDA in response to Dr. Donoghue's overtures about this issue. My submission, it's clear from this statement that Highland and Travenol had previously manufactured coagulation factors from plasma obtained from prisoners. The statement doesn't define which concentrates were produced this way, but there's no suggestion that prison plasma was intentionally excluded from any particular products or that it was not used in particular markets, such as the United Kingdom. The date of that document is 20th of October, 1983.
On the 24th of March, 83, which would be some 7 months earlier, the FDA had recommended to pharmaceutical companies they shouldn't make any more product from those donors thought to be in a high-risk group.
That's correct, sir.
It doesn't say whether prisoners in the United States penal institutions were thought to be members of a high-risk group, but it's implicit in this document that at least by October they were thought to be a riskier group from which to take plasma. But it looks as though between March and October they went on doing it.
Yes, sir. I think that is the only inference that can be drawn from the document. The reference to the fact that the discontinuation hasn't taken place as of the 20th of October, but is due to take place in that week.
They say in terms that they've got some in the pipeline, they're making product from it, and they're going to market it.
Yes. It is still ongoing at that time.
Yes.
The March FDA regulation recommendations are something that we will come back to.
Yes.
There is evidence that in the 1970s, objections were raised to the practice of using prison plasma. Edward Shambrom was a doctor who worked for Highland as medical director and director of research between approximately 1965 and 1972. On the 30th of October 2002, he gave a deposition as part of some litigation in the United States. And I'd like to turn to that now, please, Shumik. CGRA 40495. We can see the first page there shows what this document is. The deposition of Edward Shambrom, MD, 30th of October 2002.
I won't go to it, but internal page 10 of that transcript contains the potted history of his CV that I have just given you. If we could, Shumik, please turn to page 7 of our electronic document. This is page 43, internal of the transcript. And I will just read through the questions and answers that were given in this deposition. Question: do you specifically recall whether or not Highland had a plasma collection operation within the Angola prison in Louisiana? Answer: yes. Question: did you become aware whether or not Highland had a plasma collection arrangement with the Cook County jail system? Answer: no. Question: other than Angola prison, were you aware of any other prison facilities for which Baxter, aka Highland, was procuring plasma directly or indirectly? Answer: I don't think so. Question: did you ever express any objection to people at Baxter about the use of prison plasma? Answer: yes. Question: did you express them to the President? Answer: yes. Question: and what was the nature of your objections? Answer: that hepatitis was present at the prison. Question: was it to your belief more rampant within prison population than it was within the general population? Answer: no, just at Angola. Question: just Angola. Have you ever had occasion to learn anything about the incidence level of hepatitis in prisons other than at Angola prison? Answer: no. Question: but did you happen to believe that at Angola it was particularly high? Answer: yes. Question: what was the, if you can recall the name of the president of Baxter at that time when you expressed these objections? Answer: I think it was both Fred McCourt and his successor shortly, their president, Norm Aitchen. Question: what was their reaction to your objections? Answer: I was fired. Question: okay, so would it be fair to say that in part of the collection of things that led to your termination involved your objections about using prison plasma from Angola prison? Answer: I'd like to think so, but I don't really know. Question: would you say that your objections were strongly expressed? Answer: very strongly. Question: and did you urge that Baxter cease using plasma collected from Angola prison? Answer: yes. It's important to note, sir, that the inquiry's copy of this deposition is incomplete, and it doesn't include any cross-examination of the witness. So this is what we would call his evidence in chief.
Yes.
I'm conscious of the time, so I have probably about 5 minutes more on prison plasma. I don't know if you would like me to continue or take a break now.
Well, let's finish the prisons before we take a break, a jail break.
If we could have, please, Shumik, CGRA 40290, please. This is a letter written on the 3rd of June 1987 from Carl E. Brooks, president of, well, it stated, it's on the heading is the Highland Therapeutics Division, Travenol Laboratories, Inc. I'm not sure if Carl E. Brooks was president just of the Highland Division or whether he was president of Baxter overall, but certainly a senior executive within the relevant company. And it's sent to Charles J. Carman, the chairman of the AIDS Task Force of the National Haemophilia Foundation. And it is in response to a television documentary which had recently been shown, the 2020 program. And we can see in the first paragraph that Mr. Brooks is saying that he's pleased that NHF recognises that the program was highly biased and distorted. I'd like to pick up the letter, please, from the third paragraph, which says this: we feel that Highland Travenol has at all times been responsible in its actions on behalf of the people who live with haemophilia.
you and the NHF must recognise that the whole treatment community shares in the responsibility for some of the situations that exist today. By demanding the lowest possible price for coagulation factors, you contribute to the need for plasmapheresis establishment owners and operators to operate in less desirable areas, prisons, and locations where plasma is available at low costs. This section is highlighted in the text. The NHF, we believe, has a clear responsibility to support organisations that act in the best interests of the patient population, even when it may result in a slightly higher cost. There has been a strong tendency over the past couple of years to support those organisations that offer the lowest cost, rather than support those who invest in improving the safety and efficacy of today's as well as tomorrow's products for the treatment of haemophilia. Over to the next page, please. Highland continues to be committed to leadership in haemophilia therapy worldwide. We manufactured the first AHF concentrate and provided the first heat-treated AHF. Highland developed the first product for treatment of inhibitor patients. Highland has invested heavily in rDNA factor VIII research and development and was the first to begin human clinical trials of that material. We were the first manufacturer to provide a quality home care for treatment of patients with haemophilia in this country. We were the first manufacturer to screen all plasma in compliance with the German BGA requirements in 1985 and behaved in a responsible fashion by not using material rejected for Germany's ALT requirements in any other market, as several other US manufacturers did. Soon, we hope to market a new high-purity, viral-safe AHF purified from plasma using monoclonal antibody affinity purification technique and improved viral inactivation technology. Highland Travenol decided prison locations were not the best for collecting source plasma several years ago and decided to cancel contracts with all prison sources. Certainly, based on information available today, no one can argue that prisons are locations where there are more than the usual population of what we now agree are high-risk groups. Further, over the past 4 or 5 years, we have either sold or closed down Highland-owned centres that are in the less desirable areas and have cancelled contracts with independent centres located in these areas or insisted on their relocation.
Just pausing there for a moment. What does that? How do you interpret the second from last sentence? Certainly, based on information available today, no one can argue that prisons are locations where there are more than the usual population of what we now agree are high-risk groups. What's it saying?
As I understand it, he is saying that the prison population mirrors the general population in terms of potentially high-risk donors. I don't know if that is a reference to levels of hepatitis C, or as it would then have been known, non-A, non-B hepatitis, whether it is a reference to levels of AIDS or HIV in the prison population. But it seems to be an argument that there is no particular risk that associates with a population of prison donors as compared to a population of donors drawn from the general public.
And it seems to lead on to the description we've either sold or closed down Highland-owned centres that are in these less desirable areas. These being a reflection back to prisons with no more than the usual population of high-risk groups. I don't quite understand what's being said.
I don't know if a reference to these less desirable areas is a reference to prisons or is a reference to the point that was being made on the previous page, which is that the desire for low-cost haemophilia product has led to manufacturers obtaining plasma from, quotes, less desirable areas. My initial reading of it was that it's a reference to that earlier point that was being made rather than to the prison group. He's saying that we've done both, as it were. We've
What do you mean, the
Stopped
point, the
collecting.
point where the author blames the customer for the lack of safety of the product
Yes,
because he wants it cheap?
yes. Yes, so the reference on the first page is to the need for plasmapheresis establishments, as the author saw it, to be operated in, quotes, less desirable areas.
Yes.
So I took that reference to, on the second page to be a comment that those plasmapheresis centres have been closed down, as well as the ones in prison.
And then you were going to turn to the last paragraph.
Yes, the last paragraph says, I trust, Charles, that just as Highland led the way in vacating prisons and will conform with your request to move our collection sites for plasma to more desirable locations, as well as refuse to use brokered plasma where quality of collection cannot be adequately assured, but we will have the support of the NHF, the treaters and the patients, even if it does cause Highland's costs to increase somewhat. I also trust that you will appropriately deal with those manufacturers who fail to comply through refusal to purchase or use their products and services. That is the letter of...
I mean, it seems to imply there that it is yet to move its collection sites to what it describes as more desirable locations, and it is until the NHF got on the case. It was using brokered plasma, buying plasma from plasma brokers.
Yes, I think...
Is there any reference in the Travenol Limited, what's the date of this, 3rd of June 87?
Yes.
The document that we saw earlier from Travenol Limited, setting out the identity of the places from which plasma was collected. Does it mention anything about buying from brokers?
I don't think that it does. I will go back and I will check that, and I will check Dr. Kingdon's statement. The phrasing of the letter is that it seems to be a response to something that has been asked of Travenol by the National Haemophilia Foundation. The refusal to use brokered plasma doesn't necessarily imply an acceptance that brokered plasma was used in the past.
Well,
It's saying that it won't...
does it not? I mean, it's just as Highland led the way in vacating prisons and leading the way will conform with your request to move our collection sites as well as refuse to use brokered plasma.
I don't read that as being an acceptance that
Of
brokered
past
plasma
use,
was...
just saying that we are agreeing to refuse to use it, but it hasn't said, as it might have otherwise said, we don't ever use it.
It hasn't in this letter, no. I will check, though, with Dr. King.
I mean, I take your point. It doesn't say in terms, we have been using it, we're now going to stop. But it's perhaps would have been more in tone with the rest of the letter if it said we led the way in not using it at all.
Yes, it doesn't say that. But again, this letter clearly demonstrates that prison plasma had been used by Highland Travenol in the past.
Yes.
That's all I was going to say about the use of prison plasma at this stage. It's, as I say, something we'll come back to in November.
Well, we'll take a break then till 5 to 12. 5 to 12.